Today's lesson is centered around things that are happening within the cell and through the cell membrane. Remember, a couple days ago, you did this handout here where it looked at active transport. In other words, the transporting of materials into and out of a cell. There were several pages to it. You actually went through the central ideas that I'll touch on today with you. That's also found in 7.4. And as you look at this, it's kind of a fun way of imagining why you drink maybe the fluids you do during sports and how it affects you during the summertime and even the wintertime. So, number one, this is our lipid bilayer. It is a phospholipid bilayer. And one of the things that we are trying to constantly do is maintain this internal stable environment in all of your cells. So, how do you do that? That's really kind of the central question. Well, there are many factors that come into play. pH is one of those, concentration or how much something is present. And we're trying to make sure that is the same on both sides of the cell membrane. So there are certain properties that the cell membrane has, as you looked at earlier in the chapter. And so 7.4 looks at how these molecules are moving across to adjust to the necessary needs of the cell. So, quick physics lesson. Remember that molecules are in constant motion. They are constantly bouncing off of each other. Solids, liquids, and gases, all three forms are constantly moving. Gases move about the most free of the three phases, but solids still are mixing. They're just vibrating but are still moving at a really slow rate. So this is the energy of motion, which is called kinetic energy, and this is a little bit more than you'll need to know. But in physics class, you'll look at a little bit more about the kinetic of motion, and kind of work through some of those problems that are kind of fun. But that's physics. So just be aware that molecules are in this constant motion, which leads us to this first phase, which is called diffusion. So for our purposes in biology, diffusion are this movement of molecules from where they're greatest, the greatest concentration, to areas where there is less of them or lower concentration. So on the diagram, you can see molecules are moving from the right side to the left side of this imaginary test tube. And the difference is that there's something in between the middle. We'll call this membrane. So in other words, the dotted lines would indicate some type of barrier that's being very selective, much like our cell membranes would do, selecting what's going to go across it. In this case, molecules will always be moving, that kinetic energy, going from areas of high to low concentration. So in this next phase, then we're going to realize that this is constantly happening. Even when they reach homeostasis or this equilibrium, molecules are still going to be moving back and forth. It doesn't just stop. So the question is then, do molecules diffuse faster in warm or cold temperatures? Well, it's kind of a funny thing, because warm actually turns out is nothing more than how fast molecules are moving. So that's what warm is. So when you're warm, that means that those molecules are moving very, very quickly. The other thing is diffusion occurs across the membrane can be determined largely by size and the charges that these molecules have. Remember when we suggested that water is kind of a unique molecule and that it's charged on one side, on the oxygen side it's negative, and on the hydrogen side it's positive. So these charged molecules, like water and other ions, are going to move across based on sometimes those charges. So oxygen, glucose, some of them, the regulars, carbon dioxide, will fuse across that membrane based on this. Proteins can't, because they're so large. And so size will make a difference. So we'll have to be able to move large molecules like proteins in a different manner. And it won't be able to squeeze through the membrane or those lipids, the lipid bilayers that we described earlier. One of the special circumstances of diffusion, however, is osmosis. And osmosis is nothing more than diffusion. And that sounds kind of strange, but is specifically one molecule, and that's water. So the diffusion of water across this membrane is what we label as osmosis. And it goes from an area of high concentration to areas of low concentration. So much like what you can see here, if I have some sugar in a beaker and the beaker is divided down the middle with some type of membrane, where there's a high concentration of this solute, the solute is the solid, in this case, the sugar, and there's a lower concentration on the other side, in this case, on the right-hand side. What we're going to see as time goes on is we jump over here, that there is going to be a movement, on this case, from one side to another. Now, the easiest thing would be just like the sugar just moves across. But what happens if it's too large and it can't? The only thing that's small enough in that case is going to be water. And that's why you see the arrows going from, in this case, the right side to the left side. In that case, what's going to happen to the other side? It's going to drop as far as its volume, and the other side where this membrane is located, the volume will increase. Hence, you're seeing the movement of the volume increase on the left side of this this fake cell, or um, this example of movement of water in this case. So that's what you're seeing right here, is just the movement of water. Maybe it's a little easier to see with this, where we have pure water on one side and salt water on the other. And if you have a semi-permeable membrane on one side, where we know that the molecules, in this case the circles, the solids, can't move from one side to the other because of maybe their size, then the only way of doing this is for the water to try to equal out the concentration on both sides by going from the right side to the left side. So I always like to look at percentages as far as the percent of the water concentration. So maybe on this side, we have a certain volume concentration of maybe 99% fresh water, 99%. And on the other side, maybe it's only 70%. And so there's going to be a shift of water going across the membrane to try to balance out for homeostasis. So that leads us to a whole host of problems or scenarios that we can look at, both with animal cells and plant cells. Let's first of all start with animal cells. With animal cells, we have a solution that's referred to as an isotonic solution, meaning in the middle here, there is equal concentration of solute or percentages of solute inside the cell and outside of the cell. So if we're looking at solution, an isotonic solution is equal. And that means that molecules of water can go into and out of the cell at the same rate. If we look at the second scenario, which is called a hypotonic solution, a hypotonic solution means that for some reason, the water concentration is lower in the cell, so water is constantly coming in. So in other words, you have a higher solution on the inside, and water is constantly trying to come in there and trying to balance that out. I always think of hypo almost like hippo. Hippos are really large around, and they're increasing as they age. Same thing with the hypotonic solution. The cells in it are going to gradually get increasing in size, like a hippopotamus would, as time goes on. In a hypertonic solution, on the far right-hand side here, hyper means just the opposite. In this case, the water concentration is higher in the cell, so the water will rush out. In other words, think of these cells like you were drinking salt water. And in other words, the concentration of salt in your blood is greater than inside. So water is going to rush out and dehydrate your cells. So this scenario right here would occur as your own body cells would be dehydrated as a result of the concentration difference between inside of your cells and maybe that's what's flowing through the circulatory system, like your blood. So kind of sum this up, if we have three different scenarios, the one right here, letter A, is a normal red blood cell. If the water concentration percentage, in other words, is the same on both sides, let's just say on the outside here it's 92% water, And on the inside, it's also 92% water. Well, that's equal concentration. Water molecules are going to go in and out at the same rate. The solutes would remain unless they can go across the membrane, which they looks like they're not, and they remain on both sides. But again, what's going to even it out is the water. In the next scenario, letter B, here we have a loss of water. In other words, the cell is becoming dehydrated because the concentration of water is greater on the inside, and therefore it rushes out to try to balance this out. Well, this is it's an endless task. It will constantly lose water as a result of this, and hence the term, the scenario of the cell is going to become losing water, and um, that's the scenario that you can see with that arrow, that strong, bold arrow leaving the cell. In the last scenario, in a hypo concentration, again, think of hippo increasing in size, the water concentration is greatest on the outside and less on the inside. So in this scenario, water is constantly going to come inside the cell. In a plant cell, it's a little bit different, where we have distilled water in like an onion cell. So the cells are stained pink. Really hard to see the nuclei here, but you can definitely see the white lines as our cell membrane and cell wall. So in this particular scenario, what you're seeing is the cells are increasing in size, and in a hypotonic solution. But the challenge is that there's a cell wall, and it's unlikely that the cells would break open and explode, or rupture. So that's what we referred to as increasing its Turgor pressure. So in other words, Turgor pressure is the concentrate, or the not concentration, but the pressure on the inside of a plant cell, and it makes it really rigid. And you can see that by the photograph. Look at the other photograph here, where it seems like the cells have actually decreased, shrunk, and are shriveling up. In this case, the same cells have been placed in a saltwater solution. So in this case, the water is leaving the cell. In that case, so the next scenario, therefore, is we're looking at the ability for that cell to lose water. It's not necessarily good for the plant, and when that happens, the cell shrinks, and we call this wilting. In other words, the plant, you've seen it happen on your decks, or if you're not watering your plants, they will just decrease their ability to hold up that particular plant in some of the flowers and or leaves. So this is a place where a cell is in a hypertonic solution. In other words, the cells aren't rigid, and in this case, they can't hold it to mass. That would be a bad scenario for the plant. All right. So in summary, then we have three scenarios for plant cells as well. Nothing different, where we have a plant in an isotonic solution. Let me just flip that back. In an isotonic solution, and in an isotonic solution, the molecules of water going into and out of the cell at the same rate. In a hypertonic solution, notice the arrows are leaving the cell. The cell is referred to as plasmonized, and therefore it's losing greater amounts of water than what's coming in. And on the last scenario, where we have a hypertonic solution, we have more water coming in that's leaving. Water's still leaving, but it's just at a lower rate, just like the hypertonic solution. And therefore, it's becoming very turgid. In other words, the pressure on the inside is becoming very great, just like blowing up a balloon. All right, a couple other elements that your chapter covers as well. And there are a couple different scenarios of diffusion. And what I wanted to bring up is the first one. It's called facilitated diffusion. Facilitated diffusion, if you recall, are about those molecules that can't go across the membrane and they need help. Now, the help's just that opening or the doorway. And so in this case, if you look at the red dot, it's maybe like a protein, that the only way it can go from areas of high concentration to low concentration is through with the aid of that protein. And that's what you're seeing here. There's no energy required. And therefore, we refer to this as kind of a passive type of transport. Passive meaning no energy is required. So that's number one. A second one if we look at this, is a type of active transport. And active transport requires energy. And so when we look at active transport, molecules again are too large to go through the membrane, and therefore, again, they're relying on something like a protein that would allow them to travel through it. So in this case, I have some hydrogen ions, same scenario. They require some type of protein in order to go from one area to another. Here in active transport, however, we're going to require energy. And so in this particular case, you can see these hydrogen ions going from a different scenario where there's a low concentration and they're going across what is called the concentration gradient to where there's a higher concentration. Now, that's a big difference. And when we do that, again, it's going from low to high, requires energy. It's like pushing up something up a hill, it requires a lot more energy versus going down a hill, we can think of as no energy is required. And going down the hill is like a passive transport, active transport going up the hill. The two examples that your book references at the end is something called endocytosis and exocytosis, in addition to something called the sodium potassium pump. If we look at just this scenario, what's the difference between passive and active? As I said, passive requires no energy. So if you look at diffusion, where molecules go from high to low concentration, or facilitated, again, same thing, going from high to low. This case, it just needs some type of protein to go across the membrane. Active transport, here you can see the difference where there is a low concentration going to a higher concentration, it requires energy. So those are the main differences. And we can see that diffusion, facilitated diffusion, and osmosis are all example of passive. And usually it's molecules like oxygen, water, and carbon dioxide, and glucose that would be going in this passive type of transport. Versus active, these are movements of molecules, maybe ions across the membrane, and they require energy. As I said, your book gives two examples of that, endocytosis and exocytosis. I want you to look at the diagrams here and notice that this is my cell membrane. It's a phospholipid bilayer on the top and on the bottom. So on the top is demonstrating exocytosis, where you have a vesicle that is lined with the same type of lipid bilayer as the cell membrane. And as it reaches the cell membrane, it actually joins to it. You can see where the vesicle, in this case, the cell membrane join. And in the third set of arrows, you can see that it's joined and then it releases its contents inside. So these proteins are now going to be released to the outside of the cell. The opposite happens in endocytosis. In endocytosis, you have these molecules that are going into the cell or being surrounded by the cell. And again, in the next set of arrows, you can see that the cell membrane is closing around it, forming this small little package. It's almost like a Ziploc bag filled with the contents. In this case, maybe it's going to consume it at a later time. So, exocytosis, proteins and other molecules secreted out of the cell. And in endocytosis, molecules are doing just the opposite, they're coming into the cell. Both require energy in order for this to work. So this is an example of active transport, where molecules are using ATP or energy for this to occur. The last one is referred to as the sodium potassium pump. You can put down your pencils. I'm not going to have you require going through the sodium potassium pump, but I do think it's important that we realize that your nerve cells work and there's a lot of energy. So as you're taking a test or you're getting ready to think about maybe a move in a chess game, this requires energy and it requires a lot of energy. And the way this works is that it's a type of active transport where you have special proteins that are going to shift ions going from areas of low concentration to high concentration temporarily. And these ions happen to be sodium and potassium. And so the name that we give it is the sodium potassium pump. This is the way your muscle cells will work and operate along with your nervous system in conjunction. It just takes a lot of energy to flex the muscle and also just for nerve cells just to work in general. The last one that your book mentions here, and again, I'm not going to have you required to know it. It's just another example of how much energy is resulting at the cost of just thinking and using your nerve cells. Is that we have molecules going from low concentration to high concentration.